Hello students, welcome to our online video tutorial class. We have already started the chapter Principles of Inheritance and Variation, and in this specific chapter we have already completed up to the mutation and the mutagen and the types of the mutagen. Up to that we have already completed. Now in this specific video I will be starting the next part, and that is the genetic disorders. First of all, when we will be starting the genetic order, before starting the genetic disorder, we need to know regarding the pedigree analysis, because pedigree is a matter, or pedigree is actually the calculating procedure by which the transmission of any specific type of the trait or transmission of any kind of the disease from one generation to the next generation, that will be studied. So for that reason this specific pedigree is very much required. So first of all, when pedigree analysis we will be starting, we need to know what is pedigree. Pedigree is a such kind of the diagrammatic representation by which we can determine the transmission of any specific trait or any specific disease from one generation to the next generation in a single family. So that will be considered as the pedigree. So pedigree is actually the transmission of any genetic trait or any disorder from one generation to next generation in a family. That is known as, and that is proposed by in a diagrammatic representation, that is known as pedigree. First of all, when we will be starting the pedigree, we need to know about the symbols, because without knowing the meaning of these symbols, obviously the pedigree analysis is not possible. First of all, in the diagrammatic representation, if you are seeing that these small boxes which are vacant, empty small box, then this symbol means this is actually considering the male. If you are seeing the vacant sphere or vacant circle, on that time obviously it will be considered as the female. And keep it in your mind, this male and female which are empty box or empty sphere, they are obviously unaffected male and unaffected female. That means they are not carrying any kind of trait or they are not having any kind of disease. That's why they are unaffected. If you are seeing this kite shaped symbol, obviously this means the sex unspecified. That means the sex of this specific individual. Obviously this individual is unaffected, but sex is unspecified. Now these individuals, if you can see, that means these individuals are deceased individual. That means these individuals have died. Okay. If you see the blackened or darkened box or the darkened circle, so obviously this individual means these are affected individual. They are having that specific trait or they are having a specific type of disease. If you see this kind of the symbol where one arrow is actually given with a small empty box or square, so obviously this means proband. Proband means if this kind of symbol will be present in any of the pedigree chart or in any of the pedigree diagram, so obviously from that specific individual the calculation, the further determination of the disease transmission or trait transmission, that will be determined. Next, there you can see the two individuals are present which are actually considered as a parent, and they have produced offsprings, two male and one female offspring, and all individuals, males, females and parents, those all are unaffected. So these are the individuals and they are present in two generations. These two are present on the first generation, parental generation, and these two are present on the F1 generation, first filial generation. Here it is actually the single bond present in between the square and present in between that sphere. It means that is mating. If the single bond is present and then it will be cut, that means the divorce has occurred. If male and female both are connected with each other by double bond, that means they have performed or they have done consanguineous marriage. That means the consanguineous marriage means the mass marriage which has done in between the relative individuals. Okay. Dizygotic twin means these twin, male and female, maybe they are based on gender they are different, but they have produced from two different zygotes. So for that reason they are considered as a dizygotic twins. Next, monozygotic twin, that means these two twins are actually produced from a single zygote. So for that reason they are connected with each other by a single bond also. And this is actually, in case of this specific symbol, you can see this specific female individual, the circle is small and this is also cut. That means it proves that this is an aborted female child. Okay. So from this specific symbols, I think the meaning of each and everything is getting clear to everyone. Now we will be seeing how many types of the pedigree can be seen. Mainly based on the type of traits, pedigree can be of four types: autosomal dominant trait, autosomal recessive trait, X-linked dominant trait, and X-linked recessive trait. So based on these four traits, four types of pedigree can be present. Now, what is autosomal dominant? First of all, dominant trait means this trait will be getting expressed in each and every generation. That's why it is commonly known as dominant trait. and when that specific trait will be getting transmitted from one generation to the next generation through autosome, then obviously it will be known as autosomal dominant. So in this case, you can see in this pedigree chart, the male and female, that is father and mother, both are affected, and when they have met with each other, obviously the offsprings what they are produced, within that one male has been produced, that is affected, but one female which has been produced, that is unaffected. How is it possible? Keep it in your mind, they cannot be recessive, as two affected parents could not have an unaffected offspring. So autosomal dominant trait when it is actually found, so if the parents are affected, there is no chance of getting one specific offspring unaffected. If only both of the parents are affected and if they are heterozygous, on that case only this specific offspring can be unaffected. Because suppose both of the individuals, both of the parents are capital A, small a. Suppose capital A is responsible for the disease or the trait and small a is the recessive allele, and both are capital A, small a. So obviously from them one progeny will be taking birth, that is small a, small a. So obviously that specific individual which is carrying both of the recessive alleles, they are not showing any kind of the trait or the disease. So for that reason, if the parents will be only heterozygous, on that case there is a chance of getting unaffected offspring, and that is also one only. If you come to the autosomal recessive trait, obviously this kind of the trait will not be expressed in each and every generation, that's why it is the recessive trait. There should be skip of generation, and it is also getting transmitted through the autosomal chromosome, that's why it is autosomal recessive. Now you can see in this pedigree chart, male and female parent both are unaffected, but after getting unaffected also one specific male offspring is unaffected, but the female offspring has got affected. How is it possible? So here it is written cannot be dominant, as two unaffected parents could not have an affected offspring. Okay, so obviously they cannot be dominant because male and female who are mother, father, they are not affected. So obviously their offspring is getting affected. That means obviously the skip of generation can be found. So obviously they cannot be dominant. Okay, and when the affected individual is coming from the unaffected parent, that means parent must be heterozygous. And obviously keep it in your mind here, if I will consider the genotype of these two individuals are capital A, small a, and small a is responsible for the disease and trait, then obviously small a genotype containing individual, that means this one is getting affected. Otherwise heterozygous individual and homozygous individuals of a gene containing, they will not be getting affected, they remain unaffected condition. Okay, so the parents should be heterozygous, and for that case, if the parent will be heterozygous, on that case only the affected individual can take birth. That is the phenomena found in the autosomal recessive disorder. If you consider X-linked dominant trait, obviously you can see in each and every generation the affected individual will be present, so for that reason it will be dominant trait. And sex linkage, here it cannot be confirmed from this pedigree chart, but if you see very carefully, you can see that the mother is getting affected, so for that reason only one son has taken birth, that son is getting affected. And when that son has got the father, and it has that specific son or the father is just marrying another unaffected female, but when that specific son is producing the next generation daughters, 100% daughters will be getting affected. That means the transmission of the trait or the disease is happening from mother to son and then father to daughter. So for that reason, this is obviously a transmission related with the X chromosome and the dominant traits. That's why the affected individuals can be found in each and every generation. That's why it is X-linked dominant. But in case of X-linked recessive, you can see carefully here that this father is getting affected, so for that reason his daughter is getting affected. But when his daughter is getting mother, on that time that female is marrying one unaffected male individual, and for that reason when the sons are taking birth, those are all getting affected. So here also sex linkage cannot be confirmed, but 100% incidence of the affected son will be there from an affected mother. Here 100% daughter will be affected from the affected father, so for that reason it is X-linked dominant, and here the 100% son will be getting affected from the affected mother. So for that reason this is considered as the X-linked recessive trait. Okay, so this is overall the types of the pedigree charts for various different traits. Okay, so now just we are entering within the genetic disorder which is proposed by the Mendel or which are following the Mendel's law. So for that reason these disorders are commonly known as Mendelian disorders. So let's see whatever the Mendelian disorders are present. First one is color blindness. What is the color blindness genetic disorder? Keep it in your mind, it is a sex linked recessive disorder. Always it is a recessive disorder, and that is carried by the X chromosome. defect is present either in green or red cone cell. Okay, so in case of these specific people who are color blind, actually they can't distinguish properly in between the red and green color. So which are having the problem in the red cone cells, they are called as protonopia, and who are having the problem in the green cone cells, they will be considered as the deuteranopia. So protonopia and deuteranopia, these are the problems. Who can't properly see the red color, they will be protonopic, and who are problem in the green cone cells, so for that reason they can't properly see the green color, they will be deuteranopic. This defect can be found due to the mutation only, and that mutation has occurred in the certain gene which is present on the X chromosome, and that gene is responsible for producing the red and green cone cell. Also, obviously due to the mutation, in the certain gene, the color blindness has been created. It occurs in about 8% of the male all over the population and only 0.4% of the female. So we can understand it is related with the X chromosome, and it is getting expressed in the male because in case of male, one X chromosome is present, another Y chromosome is present. Y chromosome can't suppress the X chromosome, that's why X chromosome will always express, and it will show the color blind gene features which is present on the X chromosome. But if you consider the female, they are having two X chromosomes, so one is dominant. If the color blind responsible gene is present in one X chromosome also, another X chromosome can suppress that, and for that reason obviously the color blind responsible gene can't express, and the females are also not getting suffered from the color blindness. If only both of the X chromosomes are containing color blind responsible gene, on that case only the female will be getting affected, and for that case the father and the father's X chromosome should be carrying the color blind gene. That means the father should be color blind of that specific female child, and mother will be carrier, like that. So the son of a woman who carries the gene has a 50% chance of being colorblind. Obviously, when a woman is having the colorblind gene in only one X chromosome, so whoever the offsprings will be getting that specific colorblind gene containing X chromosome, they will be getting affected only. Remaining, they will not be getting affected because they are getting the fresh wild type X chromosome. So there is a 50% chance of getting colorblind. The mother is not herself colorblind because the gene is recessive. Obviously, in one X chromosome that colorblind gene is present, but in another X chromosome the dominant wild type gene is present. So obviously the dominant wild type gene will suppress the recessive colorblind gene. So for that reason, that mother will not be diseased, that mother will not be suffering from color blindness, that will mother will be carrier for the disease. A daughter will not normally colorblind unless her mother is a carrier and father is a colorblind. Obviously, when the daughter is colorblind, that means both of the X chromosomes are containing colorblind gene, and one of the X chromosome has come from the father. So for that reason, father is getting colorblind. Another X chromosome has come from the mother. But in case of that mother, one X chromosome may be containing that colorblind gene, but another X chromosome is wild type. So for that reason, mother will be carrier. Okay, so I think the color blindness concept is very much clear, and how the genes are getting transmitted and carrying the color blind gene to the next generation, and how the next generation offsprings are also getting affected by this color blindness, I think we have understood. Keep it in your mind, the mutation in some of the genes which are responsible for producing the cone cells present in the X chromosome, they are actually causing the color blindness. Next. Next we are coming hemophilia. Hemophilia is actually another type of the sex-linked recessive trait or the disorder which get transmitted from the unaffected carrier female to the some of the male progeny. Obviously, this is also getting transmitted by the carrier female, because in case of female, if the one X chromosome is containing the hemophilia gene, also another X chromosome is containing the wild type gene, and that will be suppressing the hemophilic gene, automatically the female will not be get affected, rather the female will be getting carrier for that disease. Okay, so from the carrier female, from the carrier mother, the disease will be getting transmitted to the male progeny. But if it will be getting transmitted to the male progeny, only one X chromosome is present in the male progeny, that will be containing that hemophilia gene, and for that reason that male will be getting affected, because the Y chromosome can't suppress the gene, that hemophilic gene present in the X chromosome. Keep it in your mind, in this disease there is a single protein of blood coagulating mechanism or coagulation mechanism that will be getting affected. Keep it in your mind, in the blood coagulation mechanism there is one protein present, that is factor number 8, anti-hemophilic factor. If that factor will not be producing properly, on that time the individuals will be suffering from the hemophilia. Due to this, an affected individual, a simple cut will, if it will happen, that will be resulting a huge bleeding, and the bleeding will not getting stopped also. The heterozygous female, that is the carrier for the hemophilia, may transmit the disease to the sons. Keep it in your mind, the females who are heterozygous, they are the carrier of this disease. They are not getting affected, but when it will be transmitted to their sons, obviously the sons will be getting affected. The possibility of a female becoming a hemophilic extremely rare. Keep it in your mind, possibility of a female becoming a hemophilic is extremely rare. If the female will be hemophilic, that means both of the X chromosomes should be containing hemophilia gene, and obviously these two hemophilia gene will be coming obviously from the father. Okay, if the father will be hemophilic and if the mother of that specific female will be carrier, on that case only it can come, otherwise not. But hemophilic individuals can't survive for a long time also, because they are having the problem of the blood loss. From the small cut also the blood can be lose so much. So for that reason, for a small injury also sometimes it can be found due to huge loss of blood, that individual is dying. Okay, so this is actually the hemophilia. After hemophilia, the next genetic disorder or the Mendelian disorder is there in our syllabus, that is the sickle cell anemia. What is sickle cell anemia? This is such kind of the disease where the RBC is getting sickle shaped, the biconvex round shaped RBC that can't keep its normal shape. So for that reason, the shape of this RBC is getting sickle shaped, and this is commonly known as sickle cell anemia. This is an autosomal recessive trait. So this is the first disorder which is recessive, but it is carried by the autosomal chromosome, that can be transmitted from parent to offsprings when both of the partners are carrier for the gene. Keep it in your mind, sickle cell anemia is not like that it will be found only for the male. When it is carried by the autosome, that means male and female irrespective of any gender, they can be getting affected by the sickle cell anemia. And always keep it in your mind, it will be transmitted from parent to the offspring when both of the parents will be carrier for this gene. Then only the son will be getting sickle cell anemia, sickle cell containing RBC. This disease is commonly controlled by a single pair of allele, that is HbA and HbS. HbA is the hemoglobin A and HbS is the hemoglobin that will be sickled hemoglobin. So keep it in your mind, HbA is the normal allelic form, HbS is the abnormal allelic form. Okay, so now if any individual is having the genotype HbA, that means that individual is normal. If any individual is having HbA HbS, that individual will be carrier for this specific disease. And if any individual will be containing HbS genotype, that means that individual is fully containing sickle celled RBCs in his or her blood. Out of this, only HbS genotype containing individual can show the disease phenotype, but HbA HbS, that is a heterozygous individual and homozygous individual, they are not showing any kind of the disease. Only heterozygous genotype containing individual, when they are going to the higher altitude, they are going to the mountain, due to deficiency of the oxygen, their RBC, 50% RBC, which is not present in sickled form, they will start getting sickled. So for that reason, that will be creating some respiratory problem in their body. So for that reason, heterozygous genotype containing individual should not be going to the higher altitude. This is the disadvantage for this sickle cell anemia patients and who are heterozygous exclusively. But one more advantage is also they are having, due to presence of the HbA HbS genotype, they are not getting affected by the malarial pathogen, that is Plasmodium, because when all the RBCs are not round shaped biconvex, 50% partially 50% is biconvex and 50% is round, so obviously 50% is sickle shaped, obviously the malarial pathogen will not attack them, so they will never get attacked, or they will never get suffered from the malaria type of diseases. Keep it in your mind, the heterozygous individuals appear unaffected, but they are carrier of the disease, obviously, as there is a 50% probability of transmission of the mutant gene to the progeny, thus exhibiting the sickle cell trait. This sickle cell anemia is actually created, why? Because the reason is in the protein chain structure. We all know the RBC is actually containing the hemoglobin protein, and the hemoglobin is actually containing 2 alpha and 2 beta polypeptide chains. So in the beta polypeptide chain specifically, the glutamic acid, the substitution of the glutamic acid has occurred by the help of valine, and that is actually present in the 6th position of the beta globin polypeptide chain. So let's see whatever the mutation has occurred, then we can understand properly. Suppose this is the DNA base sequence GAG in the template strand and non-template strand, that is the CTC. So from the normal, when the mutation has occurred, this GAG has been changed into GTG, okay, in the sickle cell RBC. So in the normal RBC, sickle cell has not produced till yet. Now when they will be producing the RNA, they will be producing obviously from the CTC, GAG will be produced, because CTC I have already told you, that is the strand by following which the RNA will be synthesized. So for that reason from the CTC, GAG will be produced, but in this case from the CAC, GUG will be getting produced. And keep it in your mind, this GAG codon is exclusively responsible for coding the amino acid, that is glutamic acid. But when it will be getting changed due to the mutation into GUG, automatically that will be coding the amino acid, that is valine. That means the glutamic acid is get replaced by the help of valine, and obviously the protein has got mutant, and due to the presence of this valine only in the place of glutamic acid, the structure of the RBC has got changed into sickle shape. Okay, so this is actually the reason behind the sickle cell anemia. The GAG present in the RNA, that will be getting changed into GUG in the RNA, and for that reason the glutamic acid is actually taking the place instead of the valine. So when the glutamic acid is actually taking the place instead of the valine, automatically what is happening? Obviously that protein is getting mutant. Now coming to the next some features regarding the sickle cell anemia. The family pedigree of the Queen Victoria shows a number of the hemophilic offspring. We all know hemophilia is actually found in case of this, and Queen Victoria is showing the hemophilia. The substitution of the amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin gene from GAG to GOG, that I have told you. The hemoglobin molecule polymerizes under low oxygen tension and causing the change in the shape of the RBC from the biconcave shape to the sickle-like structure. So here the what has happened in case of how the sickle cell RBC has been produced from the normal RBC, that is clearly written, as well as Queen Victoria is actually the carrier of the hemophilia, that is also mentioned here, and for that reason whatever the sons have taken birth, the princes who have also actually taken birth from Queen Victoria, all are get affected by the hemophilia disease. Okay, now just we are coming to the next disorder, that is the phenylketonuria. This is an autosomal recessive trait, obviously autosomal recessive trait. Now in this specific trait, what you can see, this is a metabolic disease also. What is the problem? In the affected individual, the specific enzyme is actually lacking, that is the phenylalanine hydroxylase enzyme, which converts commonly the phenylalanine to the tyrosine, and due to the deficiency of this enzyme phenylalanine hydroxylase, phenylalanine gets accumulated and it is getting converted into phenyl pyruvic acid. Okay, so it will be getting converted into phenyl pyruvic acid because phenylalanine is getting accumulated, and accumulation of this phenylalanine in the accumulation of this phenyl pyruvic acid in the brain, it causes the mental retardation also, that is very much important, as well as the important point is it is also exerted through the urine because of its poor absorption by the kidney. So the phenyl pyruvic acid which has been formed from the phenylalanine, that will be releasing through the urine because it is not getting absorbed properly by the kidney. So this is actually regarding the phenylketonuria, we need to keep it in our mind. After phenylketonuria, there is last Mendelian disorder is coming, that is thalassemia. We all know this is also an autosomal recessive blood disorder, so that's why it is found in case of irrespective of male and female individual. The disease transmitted from the both of the unaffected parents to the offspring. Both the parents will be carrier. On that case only the individual offspring will be getting thalassemic. So if the parents will be heterozygous, on that case only this problem can happen, because maybe the parents are carrying that specific gene within one of the chromosome, but they are not getting affected. This defect happens due to either mutation or deletion, which ultimately results in the reduced rate of synthesis of one of the globin chain, A or B chain. So thalassemia is actually of two types, alpha thalassemia and beta thalassemia. Alpha thalassemia means if the mutation has occurred in the alpha globin gene, so obviously there should be a mutant product will be coming, that is alpha globin polypeptide chain, and if the mutation has occurred in the beta globin gene, automatically the mutation product has come into the beta globin protein. So due to the mutation, actually the patient may suffer from the alpha thalassemia or rather from the beta thalassemia. And keep it in your mind, when alpha and beta polypeptide chains are not forming properly, automatically abnormal hemoglobin molecules will be getting produced, and that is resulting into anemia. Thalassemia can be classified based on which chain gets affected, alpha and beta thalassemia. So obviously thalassemia can be of two types, alpha and beta thalassemia, based on the which chain is getting affected or mutated. In alpha thalassemia, I have already told you, alpha globin chain is not formed properly due to the presence of mutation or deletion in the HBA1 and HBA2 gene present in the 16th chromosome. So in the 16th chromosome there are two genes are present, HBA1 and HBA2. Due to mutation, due to deletion present in that specific gene or occurred in that specific gene, the alpha polypeptide chain has not been produced properly and the individual is suffering from the alpha thalassemia. But another case, in case of beta thalassemia, obviously the beta globin chain has not formed properly because the mutation or the deletion has occurred in the HBB gene, which is present in the 11th chromosome, and for the mutation or the deletion in the HBB gene present in the 11th chromosome, obviously beta globin molecule, beta globin chain is not formed properly and that individual is suffering from the beta thalassemia. So this is actually total about the thalassemia. I think there's all these Mendelian disorders, whatever I have told just now, I think we have all understood these points properly. Now we will be coming to the next type of the disorder except Mendelian disorder, that is a chromosomal disorder, because these disorders can be found in the chromosomal level. Chromosomal disorder can be of two types, where the chromosome number is increasing or reducing. First of all, there is a problem, that is aneuploidy. Aneuploidy means if the chromosome number will be increasing one by one in total set, then the problem will be commonly known as aneuploidy. So the aneuploidy can be of various types. Suppose if the chromosome number will be reducing 1, then the chromosome number will be coming 2n minus 1. So it will be considered as monosomy condition. If the chromosome number will be reducing 2, then the condition will be coming 2n minus 2. So it will be considered as a nullisomy because the result is coming 0 if you are placing the value of n 1. If one more chromosome is getting added, that is 2n plus 1 condition, that is called trisomy condition. And if that two more chromosome will be getting added with the normal chromosomal configuration, then it will be coming 2n plus 2, that means tetrasomy condition. So aneuploidy is actually overall the condition when single chromosome or more than one chromosome will be getting added or will be getting subtracted from the actual set of the chromosome, that is called as aneuploidy. And euploidy means when the chromosome number will be getting enhanced or decreased also in a entire set number. Suppose in any of the individual the chromosome number is 2n, so from the 2n if it is getting 3n, one more set has getting added entirely, or it is getting 4n. So on that time this kind of problem will be, or this kind of disorder will be considered as the euploidy condition. Okay, coming to the disorders related with this chromosomal disorder, first one is Down syndrome. Down syndrome is exclusively the genetic disorder which is actually occurred due to the presence of a single chromosome or extra additional chromosome in the 21st autosome. So that's why they are commonly known as trisomy 21. This syndrome was first described by the Langdon Down. So from his name only the syndrome name has been given, Down syndrome. The affected individual is short statured with small and round head. The tongue which is present, that is furrowed tongue, and open mouth also that is partially open mouth. Keep it in your mind, their palm is broad with characteristic palm crease. Palm crease means the wrinkles are present so many on the surface of palm. Physically, psychomotor and mental development is retarded. So these developments are getting retarded. Automatically they will be suffering from the Down syndrome. And actually the reason of the Down syndrome is trisomy 21. That means addition of extra chromosome in the 21st chromosome. If you are coming to the Klinefelter disease, Klinefelter disease or Klinefelter syndrome is a genetic disease which is also caused due to the presence of an additional copy of extra X chromosome, resulting the configuration of 47 plus XXY. So these individuals are exclusively male, but with that one extra X chromosome is getting added with their genotype. So automatically they have got or they are suffering from the Klinefelter's disease. In this disease, such an individual has overall masculine development. However, the feminine development, that is gynecomastia, also can be expressed. With the masculine characteristic feature, few of the feminine features also can be found in this specific males. And these individuals are fully sterile. Keep it in your mind, because some of the female hormone levels will be increased in this specific individual or is more in these individuals. So this is actually the symptoms of the Klinefelter's disease. And if we have come to the last syndrome, that is Turner syndrome. In case of Turner syndrome, keep it in your mind, it is mostly found in the female, and due to absence of one X chromosome, their genotype is coming 44A plus XO, 44A plus 44A plus XO. That means total 45 chromosomes are present, one X chromosome is absent. So due to absence of one X chromosome, obviously they are female, but some male features are also can be found in case of this specific female. Their menstrual cycle is getting irregular, the musculature development can take place. Acha. Next is that secondary sexual characteristics also they are lacking. Their ovaries are also present in the rudimentary condition, proper development, proper growth of the ovary also has not taken place, and they are also sterile, these females. So these are actually the symptoms for the Turner syndrome females. So these are the three different types of syndromes or diseases which are actually related with the aneuploidy and euploidy condition, and these syndromes already we have discussed. So with this specific syndrome, this chapter is getting finished. So thank you everyone. I think you have studied the previous videos also, you have seen those previous videos, and please see this video, watch this video to get proper concept or the regarding the last part of the chapter regarding various kind of genetic diseases. So thanks a lot. I think you have understood this video and you have enjoyed this video a lot. Thank you everyone for watching this video. Thanks a lot.